Hi, I'm Masuda. I'm a graduate student in Rutgers University. I'm in the cellular and molecular pharmacology program, and I received the predoctoral fellowship from the PhRMA Foundation in drug discovery. Right now, my focus is on developing cancer therapy, and this is kind of from a personal motivation. I had an aunt who I lost because of cancer, and I have seen her gone through hardship because of this cancer and how the chemotherapy had adverse side effects. So, uh, for me, I was thinking like, is there a better way that we can improve the therapeutic efficacy and also can we reduce the side effects? That motivated me to my current research focus, developing a cancer therapy that would be more selective towards the cancer cell and that would be better efficacy. So currently I'm working on developing a combination therapy for the cancer treatment. You can think of cancer cells as a fast-growing city where they are trying to building up as much as possible. If we can cut down the supply of these building blocks, like nucleotides, which are essential for building up the DNA of these cancer cells, the cancer cells, they cannot construct their building anymore. If we can also cut down their maintenance, when their maintenance is not there or the repair system is not working, it will ultimately lead to collapse of the city. This will lead to the cell death. Currently, I am focusing on how we can combine these two processes and have a better therapeutic. The main tumor type or cancer type that I am currently working on, it happens because of a dysregulation in a protein complex called mTORC1. So what happens in normal cells, this mTORC1, it controls the cell proliferation and cell growth. But when it becomes cancerous, it is hyperactivated. The hyperactivation of this mTORC1, it leads to downstream activation of metabolic pathways like nucleotide synthesis upregulation. So we are thinking if we block this pathway, cell will not have enough nucleotide pool for synthesizing their DNA. But these cancer cells, they can actually upregulate their DNA damage response pathway. So if we are targeting those pathways as well, we would see that they are not regrowing. We are hoping that by combining this two strategy, we'll be able to have a better therapeutic treatment to stop the regrowth of the tumors. The one moment I would like to share is that when I was learning for this mouse experiments to do my in vivo study, the first day when I started working for the mice training, I was quite nervous and I was like standing far away from the mice cage and the mice, I was just seeing the mice roaming around and jumping around. I was quite scared. I was thinking the mouse is kind of controlling me. I could not control it. But later I figured out, okay, with time when I get more confident, I can actually do it. So now I can control them. I'm really a nature loving person. So whenever I get some time, I'll be exploring new places. And after coming to USA, it's been 3 years almost, and I have already covered more than 10 national parks. Recently, I have been to this Yellowstone National Park and also Glacier National Park. But for me, I personally like this North Cascade one. It was like the high mountains. It was so fascinating to me. It makes us remember we're so small in this whole world. For my younger self, I would say be more confident and try to accept the failure of the experiments. Don't be harsh on yourself. So failures is a part of the process. So you just have to enjoy the ride and see what's coming in front of you. I'd like to see that my work has been translated to the patients and patients are they have better treatment efficacy and there is less side effects of the cancer patients. So I can do it in both, it can be academia or in industry. I'm happy to do it either way. And I also like mentoring. So I think as a part of being a researcher, it's our responsibility to teach or mentor the young scientists so that they can also grow up and be a community to win this battle against this cancer.